It's part two. It's the muscle tissue, and we're learning all at this particular time about skeletal muscle tissue. Page 279. We just took a look at this fabulous artistic rendition, which is to a certain degree science fiction, only because it runs from gross anatomy all the way to molecular anatomy. So so that's the deal. And I alluded to the fact that the muscle cell actually, do you see that it has more than one nucleus? That's sort of like, whoa. In fact, it's got actually hundreds or thousands of nuclei. And it's because it's made up of lots of cells. And they all fused together into a single cell, and they formed what is called a syncytium when many become one. Okay? So that's the nature of a muscle cell, also called a muscle fiber. Okay? Good to keep in mind. Alrighty then, here's one now, a muscle cell. And in this particular little section here, we're having a nucleus here, a nucleus here, a nucleus here. Now, wherever we have a muscle belly that does contraction, we have muscle cells that run the entire length of the muscle belly. Okay, now if you look down at your thigh, and you look from the top of your thigh all the way down to your knee, there are some muscles, particularly sartorius, that actually has a muscle belly that runs for, goodness, it could be a couple of feet in some individuals who have really long thighs. Okay, but certainly a gross anatomy distance. Are you with me on this? This is a cell. It's microscopic, but it actually runs in its length for more than 18 inches in most adults if you're looking at the our sartorius muscle. So that's a lot of, how shall I say, fused together cells, each of which has contributed a nucleus. Okay, so I need you to sort of meditate on that and be one with it. Okay, so and then we have the cell membrane out here. Now they've decided that they would like to give the cell membrane its own name. Okay, it's why do they do that exactly? My question exactly. Why do they do that? But it's the cell membrane, and we're going to call it a sarcolemma. Okay, just because we can, just because it's muscle and not some other cell. Okay, it is exactly a cell membrane, and we're going to exactly call it a sarcolemma, spelled sarcolemma. Okay, so also these are very energetic units here. Of course, we use them for contraction and movement and all kinds of stuff. So you would expect to be seeing mitochondria, and so if you look really closely there, they have actually, it's hard to make it out, but the artist is attempting to represent mitochondria there. That's their idea of a mitochondria. Okay, so the other thing is that we have these longitudinal little devices here, these long little sort of number 2 pencils that are called myofibrils. And do you see that they're all packed together inside one cell? Okay, all of these. And again, they're running that length. Isn't that crazy? We have this enormously organized little unit here, and this is molecular. This is like subcellular. And it's actually running for like 18 inches. It's like, who'd have thunk? Okay, so, and we're going to look at a little section of that here. We're going to enlarge it, take a look at it, check it out. So here we are. We have this we have this situation here where we have some sort of thick filaments and we have some thin filaments. Okay, and do you notice that the thick filaments are hooked up over here? They're just actually kind of hooked up right here. And the thin filaments are running along this little Z line here, or they call it a Z disc. I like to call it a Z line. But anyways, the thin filaments are hooked up to the Z unit right there. And they're sort of interdigitating with each other. See, they're sort of sliding together. Okay, so you can you can actually measure distance this way between two Z lines or between two H zones, that sort of thing. So here we're going to actually look at a particular unit here from one Z line to the next Z line. Are you with me on that? From one Z line, I need to get a little sip here. One Z line to the next Z line. Okay, so we have quite a story to tell with this now. I'm going to introduce you to the players, and some excitement is going to ensue here. I think you'll like it. Okay. These thin things here, the thin filament is the actin, and the thick filament is a bunch of packed together myosin filaments. Okay. And so that's our situation here. Notice that the myosin is sort of hooked up to a spring-like device here, and that the actin doesn't have that going on, and they're separate from each other, separate. Okay, just the way you like it. Okay, now there's going to be a story to be told here because there's actually something that's covering the actin, sort of like a little bikini, as it were, right? It's sort of like lingerie for the actin filament here, and it's covering a little what we would call a binding site. So at every little turn of the helix, do you see that this is a double actin filament here, and it's sort of spun into a helix? There's two of them, and at every little turn here, we're going to be having little binding sites, and here's the myosin heads that would like to poke up in there and bind to those sites. Okay, it's somewhat, how shall I say, ribald, but nevertheless, the actin actually has its own little covering, right, so that it's protected from that event. Okay, so that's our deal. We'll come back to that picture from time to time, or actually, they provide it for us on multiple occasions here. Okay, so when we're going from Z line to Z line, let me just retrace my steps here for one moment. Let me just sort of explain. I'm going to sort of do a little spoiler alert here. I'm going to tell you sort of like the end game of what's actually going on. Now, you can't see the binding site. Sometimes they actually color it a special color. But what happens if the myosin head binds to a binding site on the actin? What happens is that the myosin head then bends. So the ones over here are going to bend this way, and the ones over here are going to bend this way. The result of that is that it's going to actually bind to the actin site many times with all of these little heads, and it's going to drag the actin this way, and it's going to drag this actin this way. Are you with me on that? And so that means that the two Z lines or discs here are going to come closer together due to that change in conformation, because the myosin head is going to bind, and then it's going to bend its little head. It's going to bind, and then it's going to bend its little head. Okay? And so we're pulling those two actin filaments together, and we're shortening the distance between the two Z lines. The name of that distance is sarcomere. Are you with me on this? Sarcomere. So from one Z line to the next Z line, we have a sarcomere, and it's a variable length. It could be all stretched out, or you could have had the heads bind and turn, and then it gets short. Now, the deal is that we have this molecular structure laid out for like inches. Are you with me? So if I were to shorten this by 10%, I would get actually a change in the conformation of the very organ that I'm considering. That organ would bunch up and contract, and with such force that it moves the bones in the joint. Okay, and sometimes it drags a few other things with it. If you happen to have something in your hand, that's going to move too. Okay, so that's the deal here, is that this is, in fact, where the rubber hits the pavement, so to speak, is where the actual action is. Okay, this is our sarcomere. Shortening the sarcomere is going to cause contraction. So we'll pick it up on the next page then.